Hello students, today we are going to discuss about an interesting and emerging blooming field in plant biotechnology called plant tissue culture. The plant tissue culture, the name itself indicates you the part of the plant tissue is going to be cultured. Coming to the objective parts. By using plant tissue culture, we can multiply more number of plants without seeds. And by using this technique, we can conserve the endangered plants available in the forest resource. Definition: it is a technique where the new plants are regenerated by a single plant pot. This plant pots is also known as explant. This technique was devised by a German botanist Haberland in the year 1902. Now, as I already mentioned you, what is an explant? Explant is nothing but a portion of tissue taken from leaf. Then it is called leaf explant. If it is a shoot tip, then the explant is known as shoot tip explant. If a portion of tissue is a root, then it is called root tip explant. Types of cultures. The types of cultures is going to be used depends upon the explant that are used. If you are using a meristem, then the culture is known as meristem tip culture. If we are going to use an organ like anther, then the culture is known as anther culture. If we are going to use an embryo, then the culture is known as embryo culture. Techniques involved in plant tissue culture. There are 3 steps are involved in plant tissue culture techniques. Namely, step 1, excising or cutting of plant pots, that is explant. Step number 2, the explant are grown in a suitable medium are called MS medium. This MS medium is most suitable for almost all the plants. The composition of the medium. Medium containing macronutrients, micronutrients, minerals, vitamins and growth regulators. Macronutrients means the medium required for large in number. Micronutrients means the amount of the elements is very less. Then coming to the growth regulators, 2 types of growth regulators are there. They are auxin and cytokinin. Auxin mostly used for root induction and cytokinin mostly used for shoot induction. The example of auxin is IAA, IBA, NAA, 2,4-D. IAA means indole acetic acid, IBA means indole butyric acid, NAA means naphthalene acetic acid, 2,4-D 2,4-dichlorophenoxyacetic acid. Cytokinin examples are BA, that is benzyl aminopurine, kinetin and zeatin. Step number 3. If the explant grown in a medium and form either a mass of undifferentiated cells called callus, nor it will produce number of shoots and roots. From the explant we can get only a mass of cells, means it is callus. This type of regeneration is known as indirect regeneration. That is, from the explant, first callus is generated. From the callus, shoot or root are raised. That is called indirect regeneration. From the explant, directly shoot or root will emerge, means this is direct organogenesis. To conclude this session, I briefly conclude with the help of the application of plant tissue culture. From tissue culture, we can get more number of plants. That is, by using a single portion of tissue, we can get more number of plants. And number 2, we can conserve and endanger plants. And number 3, the tissue culture technique is mainly helpful in agriculture, floriculture and horticulture. Thank you.